So, good afternoon, everyone. We hope you're doing well, and thank you for joining us today. My name is Sangya Pundir. I work at the protein function team, which is responsible for UniProt at the EMBL-EBI. But all of the consortium has helped put out this portal and indeed helped put up these slides for you today. So let's begin. UniProt, also known as the Universal Protein Resource, is a comprehensive and high-quality, freely accessible resource of protein sequence and functional information. It is created by a consortium, a collaboration of 3 institutes: the European Bioinformatics Institute, EMBL-EBI at Cambridge in the UK, the Protein Information Resource, or PIR, in Washington, DC and Delaware in the USA, and the Swiss Institute of Bioinformatics, which is SIB at Geneva in Switzerland. UniProt provides not only protein sequences, but also expertly annotated as well as automatically annotated data, which describes the biology of proteins, such as the diseases they might be involved in, the reactions they're involved in, their pathways, interactions, and so on. UniProt.org provides large amounts of protein data set for consumption by the community. Indeed, it is the leading source of protein sequence and functional information in the world. And the chief amongst these data sets, which is perhaps the most important and the most used, is the UniProt knowledge base, also known as UniProtKB. UniProtKB is further divided amongst 2 data sets. As you can see here, SwissProt, which is expertly curated, expertly annotated by our curators, and Tremble, which is automatically annotated by rule-based and machine learning procedures, and has not yet been curated by an expert curator. A Tremble entry, which is picked up and reviewed and curated by a curator, becomes part of SwissProt. That's sort of the life cycle of data within UniProt. And all of this is available on UniProt.org. And one key highlight about UniProt, or point to mention relevant here, is that UniProt is released every 8 weeks online, which has led to us creating a new portal for the SARS-CoV-2 proteins and their receptor proteins to be able to bring these to the community before the 8-week release happens, so that we can provide pre-release data as soon as it's available to help the community in this time of the COVID-19 outbreak. You can access this portal through this big red button on the homepage right here, or clicking on the image, and these will bring you to a page that looks like this. This is the UniProt COVID-19 portal. Data that is available through this portal is also available through an FTP link, as shown here, which is also shown on the page itself in the message section here. So to describe why we created this page, as I mentioned, this portal provides the latest version of pre-release SARS-CoV-2 and receptor protein entries in UniProtKB, which have not yet been made available through UniProt.org. These will be updated as soon as new data or annotations are available, as soon as new sequences come in or our curators update these entries, independent of the general UniProt release cycle. For all other entries and functionality, if you're interested in looking at them, you org. And also, as this is pre-release data, there might be some elements such as cross-reference links that might depend on syncing with other databases that link to us and that we link to, and those will still largely be based on the normal release cycle. So you might see more cross-references on UniProt.org that will become available on this site as well as releases happen. This is the latest version of the data available for the entries that this portal provides. So taking a step back from the portal, let's talk about the COVID-19 disease itself. As we know, it is caused by the SARS coronavirus 2, also known as SARS-CoV-2. And this is part of the coronaviruses family, which cause illness in humans and animals. 7 different types have already been found in people, including those responsible for COVID-19 and the SARS and MERS epidemics previously. Coronaviruses, as you can see in the diagrams shown on this slide, courtesy of Viral Zone at the Swiss Institute of Bioinformatics, coronaviruses are spherical and coated with spikes of proteins. These spikes help the virus bind to and infect healthy cells in its host. The SARS-CoV genome, as you can see in the second image, is arranged in the order of 5' to 3', going from 5' with the replicase of 1/AB proteins, followed by the structural proteins, denoted here as the spike S, envelope protein E, membrane protein M, and nucleocapsid N. The SARS-CoV-2 genome has the size of 30 kb, which is generally with the size of 30 kb. Coronaviruses in general have the largest RNA genomes known to date. The genome encodes a polyprotein 1A that can be elongated by ribosomal frame shifting to produce polyprotein 1AB, both of which you can see in this image here. The short and elongated polyproteins contain 11 for PP1A and 15 for chains for PP1B respectively, and are dedicated to viral RNA transcription and replication while controlling the host antiviral defense. It is at the level of spike proteins S and NS8 that SARS-CoV-2 diverges from SARS, differing in both the amino acid sequence as well as the glycosylation. The spike glycoprotein comprises of S1 and S2 subunits, and the SARS-CoV-2 interacts with the same host receptors as SARS, such as the human protein ACE2. Taking a look now at the coronavirus polyproteins, as you can see on this image here, the large replicase polyproteins PP1A and PP1AB, encoded by the partially overlapping 5' terminal of 1A and B, within the 5' 2/3 of the genome is proteolytic cleaved into 16 putative non-structural proteins known as NSPs. These include 2 viral cysteine proteases, a papain-like and a chymotrypsin-like protease, also known as the 3C-like or main protease, an RNA-dependent RNA polymerase, NSP13 helicase, and other proteins which are likely to be involved in the transcription and replication of the virus itself. Looking at the SARS-CoV-2 subgenomic RNA, structural proteins are made from a nested set of subgenomic mRNAs with shared 5' and 3' sequences. These mRNA are produced during replication on specific sites by a translation of all ORF in the 3' part of the genome. These include all the virion proteins and host isolation proteins. So, having had a quick briefing of the biology behind this data, let's look at how you can explore the SARS-CoV-2 and receptor protein data through this new portal that we're here to look at. You can click on the link if you've downloaded the slides, or go to the UniProt.org homepage and click on the red button. It's also available on covid-19.uniprot.org. And if you click on the page that looks like this, I believe everyone can hear and see me well enough. A quick check on the chat box. There are no issues. Okay, it looks like it works. Looking at the page you will land on, which is the COVID-19 disease portal for UniProt. As soon as you are here, you will see a message that describes that this is the pre-release latest version of the dataset available for SARS-CoV-2 and its receptor proteins. And hence, this might be different to the data you might see on UniProt.org. This data will be more up to date, as UniProt.org is only updated every 8 weeks, whereas this site will be updated as and when updates become available. You can also access this data through the FTP. And as I mentioned previously, if you would like to look at other entries, you can go to UniProt.org. So focusing on the functionality here, firstly, there is a search box on top of the page. And just to mention, this search box only searches within the protein entries available on this portal. At the moment, there's 16 protein entries available here, 14 of which belong to the SARS-CoV-2 virus, and 2 of them belong to the human receptor proteins. So you can search within them, and you can also filter within them using the filters on the left-hand side of the page, which can give you a quick snapshot of the data as well. You can see that 15 of these are expertly curated, and 1 belongs to Tremble, which is automatically annotated. You can see the organisms that these proteins belong to, and different you filter by different attributes, such as whether or not they have a 3D structure and so on. You can also click on this arrow sign next to the page title to collapse the message if you'd like to expand the area of the page where you can see the results like this. Looking at what else you can do on this page, there is a download button. So if you'd like to download all of these entries, you can do that straight away in one of the various formats that we provide. If you choose to download these in one of our tabular formats, such as the tab separated or the Excel formats, you will also be given a choice of which columns you would like to have in your download. And it'll look a bit like this. You can see the columns that are part of the default set, and you can search for other columns that you might think you might be interested in, such as function or other names. You can also, if you don't know of what the column names might be, you can also browse the column names by clicking on the different sections here to select them or deselect them. There is a number on the very right-hand side of these sections right before the arrow that shows you how many columns of a certain category are in your selection. You can also see them up here. You can drag and drop them, and then you can download them in your chosen format so that you've created the data set that you're interested in with all of the columns you would like to have. Going back to this page, you can also choose to view this information in 2 formats. You can see here the information in the form of info cards, which give you the accession and the entry name, the protein name, the species name, the gene, the length of the sequence, and some keywords and highlights about the protein entry itself. If you are more familiar with the conventional UniProtKB results page, as seen on UniProt.org, you can click on this button here, which will switch you to a results table that looks a bit like this. And you can still customize this results table to change the columns you are seeing and add more by clicking on the customize data button here, which will give you the same kind of customization wizard that I described earlier. And click on the download button here and download in one of the various formats that we provide. The other thing you can do, of course, is click on one of these links and go into the entry itself to view all the details and all of the annotation we provide for it. And that page will look a bit like this. On top of the page, you've got the introduction panel that gives you the kind of names and organism information for this protein, evidence for its existence, and annotation score. And as you scroll, you will see all of the curated information available. You can see the function heading just in this screenshot, but on the left, you can see that you can also jump to the different sections, providing names and taxonomy after function, subcellular location, protein processing, interaction, structure, family domains, and sequence. And other sections are grayed out here, but as information becomes available, they might also be accessible to be clicked. So on this page, you can again download data straight away in one of the formats available, or you can scroll through and view data. Thing to mention, if you scroll down to function, you will notice that there are evidence tags at the end of the content that show where the data comes from. And you will see a lot of them saying by similarity at the moment, as a lot of the curation is currently being done based on sequence analysis and similarity to other SARS virus proteins that we are familiar with. As more publications and information becomes available about SARS-CoV-2, specifically, this data is also being curated and added to this portal and to UniProt entries. And you will then start to see more types of evidence tags. If there are publications linked to some information, you will see them shown here directly and linked from these tags directly. So scrolling down, you can see more information such as catalytic activity, as well as sequence positional information. So information that's specific to certain parts of the sequence, such as the cleavage sites and so on. They look like this. You can see on top there is a scale showing which part of the sequence you're looking at, and a visual representation of the sequence features specific to those positions. You can click on these, such as let's click on this rectangle here, and that will highlight that feature in the table attached to this visualization. And it will also highlight any other sequence features that might be overlapping exactly the same position, so you can see exactly what's happening at the position of the feature you're interested in. And again, there are evidence tags showing the source of the information here. Scrolling on, you will see other sections such as names and taxonomy, which provides the recommended and short protein names and so on. It also provides information about the proteome that this protein belongs to. Just to highlight here, there is an identifier available for the proteome already for the SARS-CoV-2 viral protein. It is not linked yet, as you can see, because we're still processing the proteome. But as that information gets added, as the proteomes get added, this identifier will be linked, and the identifier is already stable and will remain as the one shown here. So the links will be added in due time. So as you scroll on, you'll see other parts of the entry. You can go down and see the sequence. You can copy the sequence. You can submit it to different tools for analyzing it. If you'd like to blast the sequence, you can do that from this page directly by clicking on the blast button here, which will launch a blast job in the UniProt.org main website for you and take you to the website to show you the results of that blast search. As I mentioned, you can download the FASTA and copy the sequence anyway to submit it to tools of your choice. Another very important element that I'd like to mention is to encourage you to help provide information to the community about these novel virus proteins. So you can use the Add a Publication button here, highlighted in red now, to provide submissions of publications you would like to suggest, as well as annotations that you would like to submit to us for these protein entries. If you click on the Add a Publication button, you will see that it'll take you to a submissions page that looks like this. You can sign in with your ORCID ID and submit publications as well as annotations for the very many different categories that UniProt provides information in. So we encourage you to do this if you come across publications or annotations you'd like to submit. If you do that, you might be interested to know where these publications will become available, or even if you haven't submitted, you might want to see what others have. So you can click on the publications tab on the left-hand side right there, and it'll take you to a page that looks like this. For this particular viral protein that I'm looking at, there's only one publication available that has been curated for it, and you can see that listed here. But we do provide publications of 3 types, ones that have been curated by our expert curators in case of SwissProt entries, ones that have been computationally mapped to this protein entry, as well as ones that have been submitted by the community. And all of those types of publications will be accessible from this page here. If community members have submitted annotations along with publications, they will also be available on this page and become visible here. So yeah, we encourage you to contribute to this knowledge, especially with these novel protein entries, and help the community as well. So that was a quick walkthrough the functionalities here. I think the only other thing left to mention is the external links, which provides cross-references. As I mentioned before, this is a work in progress. We are still mapping, as these entries are very new and haven't been released yet, we are still mapping data with other databases. So more and more links and cross-references will become available in time. Which brings me to our future plans. In terms of curation, we are working on adding 3D structured data as it becomes available through PDB. We are also working on adding protein interaction data in collaboration with the IMEX consortium that UniProt is active within. And we are working on adding access to protein complexes data through the complex portal by adding links to these. And of course, we are curating these protein entries as a priority as any more information and publications become available. In terms of future development plans for the functionality available on the site, we are working on adding the full ProtVista positional sequence feature viewer, which looks like the snapshot you can see here on my slide now. This is currently available on UniProt.org on all of the entry pages through a link on the left-hand side of those pages that says feature viewer. If you click on it, you will see this ProtVista visualization for all of the protein entries in UniProt, and we will shortly be adding this also to protein entries in the COVID-19 portal here. This will allow you to view all of the overlapping sequence features and analyze them and find out any patterns of co-localization that might be interesting. We will also be updating the portal with an interaction viewer, so more visualizations to come in general. And as I mentioned before, we are connecting and integrating data for COVID-19 with other bioinformatics resources, such as working to link to structures and electron microscopy data from PDBE or EMDB, and drugs and small compounds binding data from Kimber, for example, and proteomics data, for example, from PRIDE. So we'll be adding data and links to these resources. We're also working on adding additional literature from other resources, and of course, researchers' contributions from the add a publication feature that might allow researchers to reach out directly to us and submit publications and annotation data. So yes, I would like to just mention again that the COVID-19 portal is released and updated as soon as new data becomes available. We are working on adding date stamps to these, so you can see clearly when data was last updated and if there's something new that you can look at. For now, I would encourage you to check it frequently. The main UniProt.org website is released every 8 weeks, but that will not be the case for the portal. So please check frequently, and we will work on adding date stamps so that you can see when the latest data was added. So that was it for this presentation. We will move on to the questions and answers now. Just to mention, we are looking for feedback from you to help us understand what you would like to see from this portal. For example, would you like other SARS viruses to be added here? Would you like other isolates of the SARS-CoV-2 virus? More proteome data? We would like to know. So there is a survey we have created, which is linked from the slides, but will also be emailed out to all of you that have attended the webinar or signed up for it. So I will send you the link. Don't worry if you haven't got the slides. I will send you the link in a follow-up email, and we would really love to hear from you. Please try out the portal and let us know what more we can provide to help you in your work. If you would like to contact us in general about the portal or any other questions, you can always write to us at help@uniprot.org. We also have accounts on Twitter, which is @UniProt, and on Facebook, which is also a page for UniProt. You can always reach out to us there as well. So that's it. I'm just going to into the chat box now and see what other questions we've got. I would just like to introduce, we also have with us various UniProt curators and bioinformaticians in the call. So if you have questions, please do ask. There will be somebody here who can hopefully answer, or at least will be the right person to hear the question. In particular, we have Philippe Lemessier, who is our virology curation expert, who has curated a lot of data. He's here to answer any questions about the data biology through the chat box, and we have various other people here with us as well. So I encourage you to please ask any questions. We will try and answer them now, or we will take them away with us. Okay, so I will just go through the chat box. Philippe, Sandra, and others, if you would like to speak up, please let me know, and I will unmute the microphones. Or if you'd like to just reply to the chat box, go for that. I'm going to just read through them. Okay, so we've had a few questions answered already. Who will verify these new annotations and publication links by Christopher? And Cecilia from UniProt has answered that there is a curator that checks the annotations and it is a live review. So you can rest assured that there will be a check on the annotation submitted through the ADA publication. So yeah, please don't be hesitant. There is a question about I can't download the materials. I will try and attach the slides to the email I sent out for the survey link to all of you. So don't worry if you can't download it now. I will send you the slides after this is finished. So don't worry about that. There were a few issues with people not having sound. I hope that that was resolved and mostly people could hear us. Okay, looks like most people could hear us. We will be uploading a link to this webinar on the portal website and also the EMBL-EBI training website. If we can get the link up before I send out the follow-up email, I will include a link there as well. So if you couldn't hear some parts of it today or we had any technical issues, you will be able to hear the webinar on the recording. We are recording this webinar. Okay, just scrolling past to see any more questions. Will this video be available afterwards? Yes, we will be. We are recording it. What information from Pride data will be added? I'm not sure specifically. I don't know if anybody else wants to chime in about that.
Yes, I can comment on that. So the mass spec peptides from proteome exchange in general, but mainly collated through Pride, will appear first on the API site. So once the Prop Vista viewer is there, hopefully we will have them visible. There is an issue with mass spec data and viruses though, because the proteome is so small, it's difficult to get statistically significant readouts from viral mass spec data, but we will do our best to get the highest quality data available to you.
Thank you, Sandra. That's Sandra Orchard, who is the content team leader for protein function content team at the EMBL-EBI, helping answer questions. Okay, so the next question is to identify the individual cleaved proteins. Is using the UniProt ID of the chain a good approach? And I believe Sandra has already answered and said that this is the correct approach, so you can try that. From Zolt, we have, why don't you have all the COVID-19 proteins in the database? I'm not sure I correctly understand the question. If you mean in the UniProt.org database, these will become available as UniProt.org goes through its regular 8-weekly releases. There are pipelines and procedures in place that mean that those releases have to be every 8 weeks, but all of the COVID-19 proteins, as they become available, will be provided through this portal. I believe we have all of them in the new portal.
Well, there's at least one open, very short open reading frame that at the moment its existence is contentious. Philippe can probably say more about it than I can, but there is one potentially one other protein, but at the moment, because there's so little evidence in it, we may include it as a tremble entry, but not as a Swiss prot.
Okay.
Well, actually, there are many small open reading frames that are in the genome, but nobody knows if it's really expressed or not. So we did not include this in the release. And we have absolutely nothing to say about this protein, no similarity to anything. So presumably not expressed. But if there are some data that indicates these are really important, we will add them up to the resource, of course.
Yes, don't forget, because there may only be 16 entries, but there are more proteins because many of these are then post-processed into shorter proteins. So count the number of pro chains, not the number of UniProt entries in the data set. So one entry may contain several post-translationally cleaved proteins.
And isoforms, of course, which are collapsed into a single SwissProt entry. So yeah, that might cover some of that.
Yeah, certainly in the human entries, there would be more due to isoforms as well. Yes, you're correct.
Okay, so as you've heard from Sandra and Philippe, there's various factors involved, but if you specifically find that there's certain proteins you're expecting to see as an entry, or you can't even find them as a chain, please write to us at help@uniprot.org, and we will speak to you and try to update the data if there are updates needed. But work is in progress to see if there's any more sequences that need to be added. Yeah, please write to us about any specifics. Okay, so looking at any other questions, I have a question from Christopher. I have a question on the subproteins, but a short chat would work. So yes, if you'd like to have a short chat, please contact us separately at help@uniprot.org. Laura is asking if I have questions on this webinar later on, which email address do I need to use? Laura, as I mentioned, I will send a follow-up email with the slides, and if I can, then with the recording, but at least with the slides after this webinar, perhaps in the next day or two, you can reply to that directly to me to ask any follow-up questions. If you miss that or don't get it or whatever happens, you can always write to us at help@uniprot.org about any follow-up questions, including any for this webinar. So feel free to do that. A couple of comments also about the missing proteins. I think that was follow-up from the discussion we were having previously. And Buds were mentioning that it would be worth it if we add all the beta coronaviruses to the portal. Thank you for the suggestion. We will make a note of that. Any more suggestions like this, please watch out for the survey link that is shown right now on my screen, is in the slides at the moment, or will be sent out in a follow-up email. We are very welcome. You're very welcome to suggest data and functionality that you will find useful. We would love to hear from you. Okay, Christopher asking for the logjam of sequences. Sorry, my scroll just keeps moving. For the logjam of sequences being determined, will you collate the variants? I don't know if anybody else wants to comment on that. Sandra or Philippe or Andrew?
Yes, and the answer is it will. We do collect variants, as I guess you are aware. There are a lot. There will be a lot for viruses, and most of them are just different strains or isolates. So in that case, collating them in a single entry is probably not the most efficient way. What we will have probably is multiple proteomes, some of which we will show in the UniProt portal, others in the UniPark. But any variants that, for instance, could bring in drug resistance, important variants, will certainly be in the entries eventually.
Okay, thank you, Sandra. There's a question from Maria saying, will there be a link for the MAB sequences related to the COV2 and structures if they are available? I believe we are in the process of adding links to structures that should become available very soon. Sandra, Philippe, would you like to add anything to that?
Well, the last paper in the pipeline already annotated, so I think within a week it should be sent to you, especially with a lot of the spike protein.
Great, thanks, Philippe. So Maria, that should be all available within a week, as you've heard. That's what we are aiming for. So please check back on the portal for that. Cecilia from UniProt has helpfully added links for the community contribution functionality I mentioned. If you would like to see examples of how those publications look, I didn't have them in the screenshot that I showed because we don't have that data on those entries, you can click on the link that Cecilia has sent. Thank you, Cecilia. Andrew mentioned from UniProt, a bioinformatician from UniProt, has mentioned that we are thinking about adding all beta coronaviruses, but wanted to know if this was useful. So as I mentioned, your suggestions are actually very useful. Please tell us what else you would like. We have made a note of this one. Christopher has asked, will each cleaved enzymic protein get an individual tremble entry? Example, the PDB sequences. Sandra or Philippe, maybe you want to answer that?
If you mean the prochains, so the mature proteins that are cleaved from the full-length transcript, Christoph, then no, they're because they're already in SwissProt, then we're more likely to retain them in SwissProt, and PDB entries will be mapped to that SwissProt entry as they come in. If we get individual submissions coming through ENA or GenBank of shorter protein sequences, then it's possible that they may end up in Tremble, but we will not deliberately create them as Tremble entries.
Okay, I hope that answers the question, Christopher. If you have any further comments, please do send them. So the next question is from Manuel. What are the main issues you have for us, the scientific community, to help with? That's a great question. Thank you, Manuel. I think for now, the main ways that we have for the community to interact with us and contribute and help is by providing publications through the Add a Publication link, as well as any annotations linked to those publications through that procedure. The other thing is, as I mentioned, we are really wanting to understand what would be useful in this portal. So if you would like to tell us, somebody has mentioned beta coronaviruses, I mean, if you'd like to see other isolates of the virus, anything like that, just to help us understand what you would find useful would be great. As I mentioned, there will be a survey going out that's linked from the slide I have up now. But if you don't get the survey or anything, if it goes missing, whatever, please email us at help@uniprot.org with any suggestions, any questions you have that might help us identify your requirements and the scientific community's requirements and try and meet them. That is actually really useful. Yes, and Cecilia has also added a manual that any feedback you have on any existing annotations on these entries, as of course on any other entries, but as we're speaking about these in particular, any feedback you have, please send that also via the help desk, which is help@uniprot.org. It's linked from every page on UniProt as well as on the new portal. So yes, we just want to, if you have any publications annotations to add, if you have any feedback on existing annotations, if you have any requests for more types of data that we really want to know, that would be fantastic. We depend on hearing from the community about their requirements and their use cases so that we can try and meet these. So yeah, yes, that's a great question, and we hope to hear from you through those channels. Any other questions? Okay, great. That looks like that's all the questions for now. Thank you to you all for attending and for joining us today. We hope you will find this portal useful. We anticipate that it will be live for the next few weeks or months as we go through these rapidly evolving times with the COVID-19 outbreak, and as information about SARS-CoV-2 virus proteins becomes more available, and as we start seeing more publications. So please check back. We will be adding more data. We will be adding more functionality. We hope you find this useful. We will send out the slides, a link to the recording if we can, and the survey link in a follow-up email to all of you. And that's it from us today. Great to see you all. I guess we're all working from home now and a little bit isolated, so this is another way to connect. Thank you for connecting with us. And that's it. We will say our goodbyes to you for now. Thank you very much.